My name is Claire Anderson. I'm a research assistant in Professor Mina Wrighton's group at the UCL Great Ormond Street Institute of Child Health. I'm going to tell you about our work using long-read sequencing with adaptive sampling that we used to discover a pathogenic repeat expansion underlining spinocerebellar ataxia type 4. SCAR4 is an autosomal dominant hereditary ataxia which is characterized by sensory neuropathy. It was first described in 1996 by Louis Tarcek in a large Utah family with Swedish ancestry. Using linkage analysis, they were able to define the SCAR4 locus within a 6 centimorgan region in chromosome 16q22. The SCAR4 locus has been further refined to a 3.7 centimorgan region in a German family. However, the causative locus for SCAR4 has remained a mystery for 28 years. A similar cerebellar ataxia in Japanese families, later coined SCAR31, has also been linked to chromosome 16, whereby a pentanucleotide repeat expansion in BEAN1 was found within that locus. More recently, a trinucleotide repeat expansion has been identified in THAP11 in Chinese families with SCAR51. Inspired by the discovery of causative structural variation in other 16q ataxias, we hypothesized that the elusive molecular cause of SCAR4 may be due to a complex structural variant. To investigate this, we adopted a targeted long-read sequencing approach with adaptive sampling on the ONT platform. This method uses real-time base calling of DNA and compares it to the target reference sequence. In this case, a 20 megabase region of interest encompassing chromosome 16q22. Sequences that do not match the reference sequence are ejected before sequencing of the strand is complete, freeing up the pore to sequence other strands, whereas on-target sequences continue to move through the pore and are fully sequenced. We performed long-read sequencing with adaptive sampling on the index cases in the original Utah family, highlighted in the pedigrees, and another affected family in Iowa, both of which have Swedish ancestry. This approach allowed the accurate detection of structural variants that may evade detection using conventional PCR-based or short-read methods, and does not require a priori assumptions about the variant type. 5 SCAR4 afflicted individuals from these two families were found to have a GGC repeat expansion in one allele of the zinc fingers homeobox 3 gene that segregates with the disease, whereas unaffected individuals have no insertion. Affected individuals were found to have between 48 and 57 GGC repeats in the last coding exon of the ZFH-X3 gene, whereas non-expanded alleles and unaffected spouses only had 21 repeats. Data from over 33,000 ataxia cases and controls also reveals no GGC repeat expansion greater than 31 repeats at this locus, which supports the pathogenicity of the expansion above 48 repeats found in the Utah and Iowa families. Therefore, this heterozygous GGC repeat expansion is rare and is likely to be linked to a founder effect within the Swedish population. Targeted long-read sequencing with adaptive sampling is a very powerful tool that we've successfully utilized to solve a 28-year unsolved conundrum. This method has the benefit of identifying complex structural variants normally resistant to PCR or short-read methods of detection without a priori assumptions of variant features. We use this method to identify a heterozygous GGC repeat expansion in the last coding exon of the ZFH-X3 gene that segregates with SCAR4 disease. SCAR4 is rare, possibly due to a Swedish founder effect, and can be characterized as a 16q repeat expansion ataxia, together with SCAR31 and SCAR51. Our work was recently published in Movement Disorders if you're interested in reading more about SCAR4 and adaptive sampling. We'd like to thank the patients and participants from Utah and Iowa, as well as the patients and participants from the 100,000 Genomes Project. We'd like to thank Professors Louis Tarcek, Ying-Hui Fu, and Catherine Matthews. I'd particularly like to thank Zhongwei Chen, Mina Wrighton, Henry Holden, Emil Gustafsson, Hannah McPherson, and the rest of the Wrighton and Holden labs.